Okay, Chapter 8. As we wrapped up last week, we finished Section 1 of the textbook and moved on to Section 2, which really concentrates a little more on human development, health, and disease. This week, we have just 2 chapters. However, this chapter that we're on right now, Chapter 8, is an extremely large chapter. There is a huge amount of information in this chapter. And it probably, rightfully so, right before a good break where you guys can have some extra time to kind of absorb a bit of this. As a reminder, watching these lectures alone is no substitute for reading the chapter and doing studies on your own. I also encourage you to use your other resources such as your Mind Brady Labs and so on and so forth. So to get started with Chapter 8, it is very likely we're going to have to chop this into a couple of sections simply for the fact that the size of the chapter is so large. So we'll get started here with the first part of human body systems. So the education standard that correlates with this chapter is to integrate the complex knowledge of the anatomy, physiology of the airway, respiratory, and circulatory systems to the practice of EMS. Of course, there's a number of objectives for the chapter. They're not a large number of them, however, they're very complex. So you can find those on 151, 152 in your textbook. Okay. Jet ahead there. All right. So, an introduction to this chapter. In order for us to know what is abnormal, we need to know what is normal. That's why a lot of the things we do in class when we're doing hands-on, we actually do focus on healthy people initially. You know, we work on our classmates starting IVs or giving injections and whatnot. And that helps us to define what we would normally expect. So when we run into those cases in which something is out of the ordinary or not the usual, we can kind of go, something's not quite right here. You may not exactly know what it is, but in most cases you can look at it and say, I know this isn't right, let's dig a little deeper. So the structures of the body are designed precisely to carry out their functions. And when we look at structure and function, we have some specific medical terms that go along with those. When we talk about the sciences of anatomy and physiology, which are not one, they're actually 2 separate sciences, but often get coupled together because they are so, they depend on each other, they're linked. Anatomy is the study of structure, so anytime that we're talking about a specific body part and how it is designed, its features and whatnot, we are studying the anatomy of it. Physiology is how does it work. So we're talking about how does your stomach work. What is the chemical processes, the physical processes that go along with that, as opposed to just, well, here's the greater curvature, here's the lesser curvature, here's the cardiac sphincter, here's the pyloric sphincter, and so on and so forth, when we can name the parts of it, but naming the parts of it doesn't always tell us the whole story of what it does. So the body has numerous layers that are involved with its normal organization. It's not just one thing. Yes, we identify the human being as an organism, but the organism has independent parts itself. I don't think that should really be a earth-shattering statement to anybody. So we go from as small as, actually even smaller than a cell we'd be talking about the chemical building blocks of the elements, and even beyond that, we could break that down into the subatomic particles of neutrons, electrons, and protons. We don't usually need to get down that far, particularly in EMS, but we do need to have a good idea of what the cell itself does. Cells will build itself up into tissues. Tissues will then form into organs. Organs form into systems, and then systems obviously become the human body. But the cell is that basic building block of life. If the cell isn't functioning, nothing else is functioning. In fact, if you want to look at how we can take a realistic view of that, the toxin that we all know as cyanide is a cellular asphyxiant. So it gets down to the cellular level, snuffs out the cells, and that's what ends people's lives. When we do something like the death penalty, those drugs that are given in Nebraska or other states where they have the death penalty, when they give those death penalty drugs, they get to a cellular level and stop all this activity. So that goes to show you that without the individual little parts of the cell, we really don't have the rest of it. We don't really need to care about the rest of it. So we are going to look at a lot of things that have cellular responsibility. All right, so your think about it actually goes back to your case study. I'll let you guys answer those questions. All right, so some general concepts in A&P and the chemical basis of life. So cells are composed of chemicals. These chemicals do a lot of things. They act as mediators, they act as building blocks, and they're obviously fuel in many cases. So we have all these different functions that these chemicals perform. Chemistry is the study of composition of substances and the interactions of various elements and compounds. Well, our body, we know the most common substance within our body, well, the two probably most common substances within our body are water and carbon, because we are a life form. Life forms on Earth are carbon-based, so we have a huge amount of carbon in our body. And then water is a combination of hydrogen and oxygen. So, and then we look at how they interact. Hydrogen and oxygen, while they do form a nice bond, they are able to be broken apart. So when we take something like water and carbon dioxide and we put them in the same environment, so water is H2O, carbon dioxide is CO2, so 2 oxygens and 1 carbon, and then go back to water again, H2O, that's 2 hydrogens and 1 oxygen. We put them in the same environment, we can break the bonds between those individual elements and combine these things into a new chemical called carbonic acid. So we watch these things and we see how does the body respond when we have these various chemicals floating around in there. And then chemistry allows these cells to engage in metabolism and to maintain homeostasis. So they're the ones that are going to break some of these things apart. They're going to create something that's usable, something that is tangible, that they can use. Otherwise, they'll turn things into waste. So I talked about that carbonic acid thing. When we have too much carbonic acid, or we have some other chemical processes that leave us lots of carbon, lots of oxygen, lots of hydrogen laying around that we don't need anymore. And anybody knows a little bit of chemistry, a little bit of A&P, or a little bit of nutrition knows that CH and O is carbohydrates. So carbon, hydrogen, and oxygen are the main building blocks of carbohydrates, which are most of our sugars. And when we have excessive bits and pieces laying around, the body can say, well, I don't need this, let's get rid of it, and it turns it into 2 major substances, carbon dioxide and water. Just like we talked about, that's what made carbonic acid. The body can do the reverse and make them back into carbon dioxide and water. And our body obviously can get rid of those in most cases by simply breathing and peeing. So to look a little bit at some structures of some atoms, don't worry, we're not going to dive too deep into this. It is, to me, it's fascinating, but I was always a science geek. I can really identify with the Big Bang Theory because, those are my people. But I never was big in physics, but biology was always my thing. But to look at these a little bit, we remember that we've got 3 major portions of an atom. We can go down philosophical roads and talk about whether we're going to be dealing with subparticles here, lower than electrons, neutrons, protons. Some people talk about quarks, and there's some plasmanates and some various other weird things, but we don't need to go down that road. When we look at a typical atom, we're looking at 3 major parts: a proton, a neutron, and an electron. Protons and neutrons are together in the center of the atom. So they make up what is referred to as the nucleus of the atom. So if you want to think about this thing maybe being more like Saturn, where you have the planet and the rings that go around it, I guess you could think of it that way. It's not technically that way, but the electrons actually are all over the outside of it. They don't go in a perfect ring, but you can think about it that way if you'd like. So the planet itself, Saturn itself, without the rings, is the neutron and the protons, and the rings themselves are the electrons. So usually we'll have the same number of protons and neutrons, and the protons are positively charged. The neutrons are without charge, or they're neutral, and then electrons are negatively charged. So this is what helps kind of keep them either separated or makes them bounce off of each other, or maybe allows them to combine. So here we see a carbon atom versus a neon atom. The neon obviously has a significantly larger amount of subatomic particles there. We've got more electrons, more neutrons, more protons than does carbon. If we want to take it to the most basic element, the most basic element we have is hydrogen. Hydrogen is the simplest atom that there is. There's one proton, there's one neutron, there's one electron, and that's all there is to it. So it's very, very simple. We can get moving up in the periodic table, and just for fun, in the course page, I'll upload a copy of the periodic table, and I have some notes on it to how it may pertain to the human body, whether it might be toxic or something that regularly found in the human body. So you can kind of take a look at that. There's a number of them that are trace elements, but depending on the day and when you look at that periodic table, you know, we're well up over 100, into the 110s, maybe even getting close to 120 elements that are found on the periodic table. And the higher you go, the more complex those elements get, with more and more subatomic particles and whatnot. Understanding that some of those elements on the periodic table on the high end are all theoretical, or have been made for split seconds. I mean, extremely small fractions of seconds when they were bombarded by something in a lab. So they're not always a completely functional and usable element, but nonetheless, they seem to believe that they exist. So some things to think about. What is a chemical element? Well, a chemical element is the most basic structure that we use in the human anatomy and physiology, as well as everything else in the universe is composed of a chemical element or combination. And what is an atom? An atom is an individual unit of that element. So it is a single, we'll call it piece. It would be like a human cell, and those atoms again have negatively charged electrons, positively charged protons, and electrically neutral neutrons. The electrical charge of those subatomic particles allows those atoms to bind with atoms of the opposite charge. So we may have some things that maybe one element has a particularly positive, a single positive charge, and another element has a single negative charge to it that has open or available. Those two can group up and work together. So some atoms are capable of either gaining or losing electrons, and when we talk about that, good examples are some things like sodium chloride. Now everybody should be familiar with sodium chloride from when we talked about the medications and the IV fluids. So the ability for sodium and chloride to bind together is because there's one atom, the sodium, which has an extra positive ion, and the chloride. All right, as I was talking about that, I recalled there was a picture of this in your textbook. Look on page 154 in your textbook. The sodium and the chloride. The sodium has this extra negatively charged electron on the outside of it. The chloride has, or chlorine I should say, has an extra space in its outer ring. Well, they like things to be all even. So when you look at the various rings of electrons, they like everybody to be full, and everybody is doesn't have extras, doesn't have spaces. And when you have this, it's easier for them to combine. So as we see, if sodium will share his extra electron with chlorine, they combine together. They can make a nice ionic compound, which fills up both of their rings and makes it so they don't have either extras or openings. And with that, we get sodium chloride, which is a fairly stable compound. And that is obviously the basis of table salt, as well as our sodium chloride solutions that we give to patients in IV form. So again, some can either gain or lose electrons. Define each of the following terms: ion, cation, anion, and electrolyte. Well, to look better at those, a cation and an anion and an ion, these are all listed right at the bottom of 153, second column, very last two sentences. You can look and see that an atom or group of atoms that carry a positive or negative electrical charge is an ion. Positively charged ion is a cation, and a negatively charged ion is an anion. You remember from anatomy and physiology, or I'm sorry, from medical terminology, we said things that have a or an are without. So don't let that confuse you and think, oh, well, it's negative, but it's without a positive charge. So the other way I think about that is there's cat, C-A-T, the T in cation. T's also look like plus signs or positives. So a cation is positive and anion is negative. And an electrolyte is a substance that dissociates into negatively and positively charged ions, so cations and anions, when placed in water. So we can dissociate these into a sodium cation and a chloride anion when we put it in water. So we can break them down, break them apart. Electrolytes are extremely important to our body. So you can see a number of electrolytes listed in Table 8.1 on page 155 in your text. Those are probably the most common, well, obviously they are the most common ions found in body fluids, and those would be, in many cases, are significant electrolytes. Sodium, potassium, calcium, magnesium, chloride, and then we have a couple of them that are still in compound form when we talk about things such as bicarbonate, biphosphate, sulfate. Very common things. Now that's not all of the chemicals that we're going to see in our body. You can see the next table there, 8.2, has some more, and we're going to actually come to that table here in just a minute. So as an AEMT, you're going to be making decisions about starting IVs and administering IV fluids. So understanding how adding fluids and solutes to the body affects the fluid balance within the fluid compartment. And so it's important to understand that. And we're going to get to that here in a minute when we talk about the tonicity, or how close to body fluid a lot of these fluids are. So we're going to get to the tonicity of them here shortly. But we also need to understand if we're putting certain fluids into a person or certain medications into a person, are we altering the balance significantly of certain chemicals or electrolytes? So if we had somebody who was who had extremely high sodium content in their body, and we were able to identify this, would it be beneficial for us to run a whole bunch of sodium chloride into their body to give them 2 or 3 liters of sodium chloride? It may not. It may actually cause some negative side effects. So kind of understanding some of these things helps us down the road when we make treatment decisions. Okay, so how do the concentrations of certain substances inside and outside the cells affect water movement? Well, that again is talking about the tonicity or the presence of certain water levels versus chemicals in the body. We're going to get to that. Like I said, I've got a great, there's a great slide coming up that's going to show that a little bit better. All right, so some of those principal elements in the body: oxygen, percentage of your body weight, 65% of it is oxygen, roughly 18.6% carbon, and 9.7% of it is hydrogen. So like I mentioned earlier, the two most common substances in our body is water and carbon. And so we have that really demonstrated here. But we also have a lot of nitrogen, calcium, phosphorus, potassium, sodium, chlorine, magnesium, sulfur, iron, iodine, and that's not all. Well, there's tons and tons and tons of other ones. But it goes on to tell you a little bit about what do some of these elements do in your body. Some of them not terribly earth-shattering, such as calcium, found in your bones, in your teeth. It's important for membrane function. So in order for our impulses to be fed from cell to cell to cell, you need to have calcium in your body to stabilize some of your cell membranes. Phosphorus a lot of times works along with calcium in your bones and your teeth, but it actually has a bigger part in the energy production within your cells. Potassium, again, this works along with calcium when it talks about membrane potential and the appropriate ability for cells to pass an electrical impulse down the line. And so on and so forth. We got lots of other chemicals there. We're going to come to those kind of on an as-needed basis. I'll back up for a second there. It would probably be a good idea that you guys start committing to memory the chemical symbols for many of these elements. I don't expect you to know the chemical symbols of the entire periodic table by any means, but the more common ones in our body, really, this is a pretty good list, just offhand. It's a pretty good list of the things that you're going to need to know. So that chemical symbol is right there between the word and the percentage of body weight. So oxygen is O, carbon is C, hydrogen is H, nitrogen is N. Now, then we start to come to some of these other ones that are a little bit more tricky. Calcium, Ca. Well, there's numerous elements that have C, that start with a C. So Ca is calcium. Where we look down the list there, chlorine is Cl. And then we have the weirdos like potassium, which is K, and sodium, which is Na. Those don't go along. Iron, same thing, Fe. Remember, these are derived many times from their Latin or earlier languages names for that certain element. So, for example, gold and silver. Gold is Au, silver is Ag. So those are another example of everyday elements that we run into that just have the weird symbols. So try to start to commit those to memory, because when we start to talk about a little bit more of the pathophysiology, we may be talking about some of these compounds inside the human body. All right, so what are the 4 chemical elements that comprise more than 99% of the body's atoms? And if we go back, we can basically roll her back to that previous chart and see it's really oxygen, carbon, hydrogen, and nitrogen. Calcium is a big player in there as well. All right, so let's talk about some of these basic basic we'll call them elements for lack of a better term, but basic substances of life. So water. Water is huge. Our body is mostly water, which is H2O. You might want to submit that to memory. H2O is water. We have intracellular and extracellular fluid. So here's a new concept for us, for those of us that haven't studied this. Intracellular would be everything that's within the cell membranes. So when we are talking about a cell, think about it as having kind of a, well, you think about a cell like this. If we take a Ziploc bag and we fill it up with water and we put some salt in there, maybe we put some sugar in there, and we'll put various other things. Maybe we put a couple grapes in there, and we put a couple of raisins in there, and we'll put some noodles in there, and a meatball, and okay, so we put a whole bunch of stuff in a Ziploc bag, and we zip it up. That's a pretty good representation of a cell. That plastic bag is the cell membrane, so it's the thing, kind of the wall that holds it all together. Cells in animals do not have cell walls. So it's an important distinction there that we make in nomenclature, and nomenclature refers to appropriate titles. But plant cells have cell walls, animal cells have cell membranes. So everything within the cell membrane, all of the water that's within the cell membrane, that is intracellular fluid. So that's the amount of water within it. Extracellular then would be everything outside the cells. We'll take, for example, just your blood. If we think about your blood, you obviously have a certain amount of water within the red cells because that's what kind of is the medium that everything kind of floats around in. So you've got water within there, but then you also have water directly outside the cell because that's what the plasma is. The plasma is really water plus proteins and a few other things, but that would be extracellular. So intra within, extra outside. So if we talk about intra and inter tend to confuse people. I think about this. We'll take example Omaha and Council Bluffs. There's a large road that runs in between Omaha and Council Bluffs and runs from state to state to state, and we call that the interstate, the interstate highway. Inter means goes between. Intra, okay, internet would be another one, goes from person to person to person. Intra is everything within. So state highways that stay completely within a state would be intrastate highways. We don't tend to use that term, but technically I believe that Interstate 1, which is found in Hawaii, should be an intrastate because it doesn't go to a different state. It only stays within one. So that's a philosophical difference, but intra would be within. So if your company that you work for has a intranet, might be stuff you can only access from work. That would be your company's intranet. So intra is within, and inter is between. Extra, extracurricular. Extracurricular would be things that you do outside of school. So intra, within, extra, outside. Now that water, we may have intravascular fluid or interstitial fluid. So intravascular would be within the blood vessels, so that would be our blood plasma. We can have interstitial, so if we roll this back one, we're talking extracellular fluids, because intracellular is everything within the cell. So extracellular we can break down again, intravascular and interstitial. So intravascular would be the fluid within the blood vessels, not within the cells, but what the cells are floating around in. And we have interstitial, which is between the cells. So I think about bricks and mortar. The mortar would be interstitial. So that's the little bits of water that kind of fill the gaps between the cells. We're going to talk about things such as solutes and solvents. I've kind of touched on these already, particularly when we were talking about medications. We have solutes and solvents. So to kind of refresh that in your mind, if we talk about, we'll take for example glucagon, the drug glucagon. There is 1 milligram in 1 milliliter of fluid, or 1 international unit. They just so happen to be the same. In this case, 1 international unit or 1 milligram within 1 milliliter of water when we're done reconstituting it. So these formed particles, so the milligrams is the solute. The solvent is the liquid we put it in. So the milliliters is the solvent. We use usually the milliliters, the solvent, in order to be able to measure it to give it. So the solutes are these formed particles, whether they're electrolytes, maybe they're carbohydrates or proteins or lipids, various drugs. That is the formed particle, or the solids. Osmosis is the movement of water across a semi-permeable membrane along a gradient from a lower to a higher solute concentration. Wow, that's deep. Okay, now is it really that deep? It's really not. We can give you a good demonstration of osmosis. Osmosis is the movement from an area of lower concentration to an area of higher concentration. So basically what it says is I'm going to go dilute you. So if I've got a whole bunch of water sitting in one spot and I drop some medication into this beaker of water, the water is going to move towards the medication. The medication is going to get then diluted out. And that's all that really is. Now, sometimes it's not that easy. And we're going to see that here in a minute when we look at this slide about the tonicity. So water and solutes move to equilibrate concentration throughout the solution. So everything in the world is working towards Zen, working towards equal and peace and harmony and all that good hippie stuff. So when we're talking about areas of higher concentration and areas of lower concentration, everything's always working to try to be the same. We've got to make it equal. So our solutes move from the areas of higher to lower concentration. And semi-permeable membranes allow water to move through it, but not the solutes. So let's take, for example, you made spaghetti. So you have these noodles that are boiling in water, and how do we finish off our noodles? We drain the water, right? So we pour it into a colander. The colander is a semi-permeable membrane. The water passes right through, or can pass through as needed, yet it retains the noodles. So our semi-permeable membrane says, I'll let some things through, but not everything. And in this case, the colander says, I'll let the water through, I won't let the noodles through. And when we have osmotic pressure, is the ability to pull water across the cell membrane. So when we're looking at osmotic pressure, it's saying, hey, I've got a high concentration in one spot. I'm going to steal you. I'm bringing you over my way because it's not equal. So let's look at this in a picture. We have 3 typical types of solutions that we deal with in healthcare. Solutions that are either isotonic, hypotonic, or hypertonic. So iso, hypo, and hyper. Iso means equal, hypo is less than, hyper is more than. So when we have a person receiving an isotonic solution, and in the case, most cases in EMS, we give normal saline, which, if you will recall, normal saline is 0.9% sodium chloride. You can get 0.45, you can get 3%, you can get all kinds of other fractions of it, but 0.9% sodium chloride is isotonic. Means that the amount of the solute and the amount of the solvent, being water, is approximately the same as most blood. So it says, when I put this in your body, we're all good. Things aren't going to go anywhere. Or if they do, they're going to do it so equally. Some of your water goes over there and some of my water goes over to you. Not a big deal. So when we're talking an isotonic solution, it just goes in there, it mixes, it intermingles, and everything is good. We don't see water shifts. When we're talking things like a hypertonic solution, a hypertonic meaning that it is a higher amount of solute and a lower amount of water, means that it's going to have to pull water to equal out. So a good example of this is dextrose 50%. D50 is a hypertonic solution. So when we put D50 into somebody's body, if it can't immediately be worked on by the blood and do some shifting and moving and dilution and whatnot, it becomes a real problem because it robs water from everywhere else around it. When we put it into the blood whisks it around and the blood mixes with it, and it pulls, it is going to pull some extra water, but nonetheless it's going to work to get it diluted out and moved around. If we accidentally give it IM, if we would give D50 IM, we don't have a readily moving flow that can disperse this stuff. So it sits there. It sits in that muscle. And because it sits in that muscle and it is hypertonic, it's going to say, all right, well, I've got a lot of sugar and not a lot of water. And the body's going to say, well, I've got a lot of water. I could use some sugar, but I have no way of getting it in because I don't have enough insulin at the moment right here. So that D50 starts to rob water from all the tissues around it, so much that it dehydrates all those other cells around it, and it actually causes those tissues to die. Hypotonic, on the other hand, says that I am such a high water, low solute mix that I am going to push my extra water off on somebody else. So we'll take, for example, the pictures that we're seeing here are examples of red blood cells. So with the isotonic solution, everything stays about the same. The hypotonic solution, which we have one we carry, it's D5W, dextrose 5% in water. That goes into the body, and the cells will rob that water. So it'll say, we need to make your sugar level equal to what we have on the inside. So we're stealing some of your water. They suck it in, causes the swell to swell up, or I'm sorry, backwards, the cell to swell up, as we see here. Or we take dextrose 50% in the third picture, so we have a ton of solute, a little amount of water, and it says, hey, I got to rob some water from you. Hey, give me some of your water, and it dehydrates. So I hope that helps. Let me give you one more example, just simply with IV fluids. If we would to give a patient normal saline goes into the vascular space, 0.9%. We put that into the vascular space for a patient who is bleeding. It stays where we put it, at least for quite some time, and it'll stay there. So it will, in theory, fill up their container a little bit more, their container being their blood vessels, fill up their container a little bit more, and help the blood vessel or the blood cells float around better. If we would give them something like D5W, let's say we're going to give trauma patients all the same fluid. Same trauma patient, let's say he's lost a third of his blood volume. We give him normal saline. We give him 2 liters of normal saline. Then that's going to help him fill up his container a little bit. We give him D5W. Give him 2 liters of D5W. Well, because of the tonicity of D5W, the cells start to rob that water. So they pull the water away. They're going to pull the water out of the vessels and either pull it into the red cells or pull it into some of the other tissues, and therefore we really don't end up making any advances because the water just all got robbed. It didn't stay in the vascular space where we needed it. And then if we would give in something called hypertonic saline, which is 3%, not 0.3, but 3% normal saline, or I'm sorry, 3% sodium chloride, the body's going to say, holy cow, there's a lot of salt out there. We need to do something about that. We're going to send the extra water. So it actually is going to create an increase of water getting shifted into that patient's blood vessels. So, but it's going to be at the expense of dehydrating some of the other cells. So we have to look at what's the best way to do this. If we're going with isotonics, it doesn't affect the other cells. Okay, so to define iso versus hyper versus hypotonic solutions. In order to define those, it's probably easiest for us to look on page 156, and we can see the descriptions of those on second column on 156. They're about halfway down. So it'll tell us that equal amounts of body fluids to solutes is isotonic. That's right in the middle of that column. We skip down to hypertonic, which is in the last paragraph there. A more concentrated solution, higher solutes, is hypertonic. And then halfway through that paragraph, a less concentrated solution is hypotonic. So I know that those are kind of vague descriptions, but hopefully talking about it by looking at the picture will help you kind of get that. So osmolarity of body fluids is somewhere between 280 and 310 milliosmoles per liter. And that's all good and fine, but in reality we're not going to know that what their normal what their current body fluid osmolarity is. But if we know that what normal is, and we happen to see this on a drug label somewhere, what the milliosmolarity of that drug is, it'll help tell us whether we're dealing with hyper, hypo, or isotonic medication. So you can look then back up at osmolarity right there in the middle of that paragraph, and talks about in terms of sodium and chloride, 0.9% solution, or normal saline, it has a concentration of 310 milliosmoles per liter, which is the top end of what our normal range is. Look something at like hypertonic, or they don't specifically list it there, but some hypertonic solutions would be above that, and hypo would be less than that. So one other good thing to remember here, and I seeing it in this paragraph here kind of jogged my memory, salt and water are best of friends. So where salt goes, water goes. So that's why people who have high salt diets and maybe are not functioning the best will often be very puffy. Their kidneys aren't working to filter out the salts. They retain the salts, therefore they retain the waters, because water is its best friend. Okay, so the sodium-potassium pump. This is where it's going to get deep again for a second here. We're looking at a cell membrane. The pinkish side of the cell membrane is inside the cell. The whitish side of the cell membrane is the extracellular or interstitial fluid, if you will. It could also be intravascular fluid. We have a phospholipid bilayer, which is the things that look like really long two-legged stick people with no arms. We have two layers of those with their legs sticking in towards the middle there. And that phospholipid bilayer is the membrane that allows some things in and some things out, keeps them out. So within that phospholipid bilayer, we have a number of receptor sites and channels that we can use to move electrolytes in and out. Now remember, water can kind of just slip through because water's the ultimate stuff. It can kind of do whatever it needs to do. It just moves right on through. But the solids of the other electrolytes don't get to slip through. So we'll take, for example, the sodium and the potassium. Sodium is Na+, potassium is K+. And what happens here is when the cell is at rest, it has a bunch of potassium on the inside. It has a bunch of sodium on the outside. As the cell's getting ready to do something, we'll say, for example, a cardiac cell. Say it's going to pass a electrical stimulus down the line. The way that it passes its electrical stimulus is changing these electrolytes, and it allows it to do its thing. Or we'll take, for example, a muscle cell that maybe we're going to contract, and the contraction obviously causes us to do some sort of work. And what happens is, as the body is starting to, the cell is starting to do whatever it is it's going to do, it allows sodium to kind of rush in because there's a small amount of sodium. Look at the picture here. The orange balls are potassium, and the blue balls are the sodium. And so the sodium on the outside is going, hey, there's not very much of us in there. And the potassium on the inside saying, well, there's not very much of us out there. But the sodium kind of is a bit more of a bully here. So the sodium then is going to push its way into the cell through a channel. And as it pushes its way into the cell to cause the cell to do something, the potassium says, well, there's not enough room in here for us, and it rushes out. Well, the body then eventually says, now wait a minute, this is not the way it's supposed to be. I need to reset. I need to put things back together the way they're supposed to go here. So the sodium-potassium exchange pump then kicks in, and what it does is it pulls back in two ions or two atoms of potassium, and it pumps back out 3 of sodium. So it resets the balance again. And you can see that they have a description, ATP in the yellow oval over to ADP, and that's describing that it takes electricity, it takes energy, I shouldn't say electricity, it takes energy for the body to do this. So the body has to break down some sugar with the help of insulin and oxygen to create power. So the sodium-potassium pump, protein on the cell membrane, keeps the ions at the proper levels inside and out. So it's a gatekeeper. It's the bouncer, if you will. The principal intracellular cation is potassium. The principal extracellular cation is sodium. So you're supposed to have, at rest, you're supposed to have lots of potassium in, lots of sodium out. When the body does something, when the cell does something, they switch places, and then it is the sodium-potassium pump's job to put them back to normal, put them back to their normal resting potential. Okay, so the sodium-potassium pump, if sodium is not removed from the cell, water will enter and in quantities that will cause the cell to burst and die. So I brought that up just a moment ago, that sodium and water are best friends. So if we have this high sodium level on the inside of the cells, they start to swell. So it requires this energy to move that sodium back out because it's still a high sodium on the outside. So it requires energy to move it back, something called active transport. And that active transport causes the body to work harder to maintain the norm. So let's take a moment to think about that concept. If you had high blood pressure, we don't even have to have high blood pressure. If we don't need them, the body knows this, the kidney says spill it, let's get rid of it. And so you pee more, and I said sodium and water are best friends. So sometimes where water goes, sodium goes. so you pee more of it off, and therefore you get rid of the extra electrolytes. Let's say your kidneys aren't working well, they're not doing their normal function, and the sodium stays within your body. So you have tons and tons of sodium running around. Let's say your level of sodium is twice as high as it normally should be within your body. So if you've got these really high levels of sodium, how hard is it now? Because we know what osmosis is supposed to do. We know what diffusion is. Diffusion is going from an area of a higher concentration to a lower concentration. So take, for example, put a drop of food coloring into a big glass of water. It's really, really dark where it hits the surface of the water. But over time, that fades away and becomes kind of a very even, pale hue throughout the entire glass. And that's diffusion. Well, if we were to try to have to put all that food coloring back into that drop, it's very difficult. It would require a lot of work to filter out the water and separate the coloring and put it back together in a little tiny drop. So that extra work is the example of what the sodium potassium pump has to do. So if you have a ton of extra sodium floating around outside the cell because all you do is eat potato chips and French fries and you salt everything, you salt things that are salted, you put this extra sodium in the body works triple time, quadruple time to try to move that. So it's that much harder on your body. And since it has to work so much harder to do it, it happens at a slower pace. And because it works at a slower pace, water leaks in to dilute the sodium. So to try to dilute that sodium, we get that water leakage, and then that water leakage becomes edema. So you get puffy, you get swollen, and because you have the extra sodium. So metabolism. Talk about a new concept of metabolism. Metabolism is the production of energy. So this is the sum of all chemical and physical changes in the body. I kind of like that. It's kind of deep. You kind of have to think about it for a second. But metabolism is all of the stuff you're doing to create energy minus all of the things you're doing to use energy. So if you're sitting around doing nothing and you're creating lots of energy, you're going to be hyper. But if you're out working, you're burning off that energy as quick, if not quicker, than you can make it. Then you have obviously you're gonna wear yourself out pretty quick, and that's a workout. I mean, it actually is now you burning the extras, the stores that you have in there. So when we look at this picture here, we can look at it and see that we have the interstitial fluid. Outside it we have some organic molecules such as amino acids, lipids, sugars, which are carbohydrates. Some of those get pulled within. They're put into the nutrient pool, so it's kind of like a picnic basket there. So the picnic basket's sitting there, and the body then starts to pull from it what it needs to do some maintenance, to grow, cause some secretion of some hormones, and it will also store some. Then the body, if necessary, if we don't have a nutrient pool, the body can then start to break things apart and use up what it has in storage. So anabolism and catabolism, which we see in the circle there, the anabolism is when the body manufactures more complex substances from simple ones, and catabolism is when it breaks them down. So it may say, we stored this as this big long chain, but now we're going to break it apart because we need parts of it to live. So in that nutrient pool, the body breaks some of that stuff down via the process of catabolism. It's usually done in a structure called the mitochondria, which we're going to get to. And it's going to create energy. Approximately 40% of it's going to be energy in the form of adenosine triphosphate, or ATP. 60% of it is going to be heat that's given off. So that's why when we're working out, we get hot, we sweat. That's why when you're in shape, even sitting around, you burn more than you would if you're not in shape. You burn more because there's more muscle there. That more muscle requires more to keep it alive, so therefore you burn more and you create more heat. So there are other things that are going to rob the body of the energy. Locomotion, contraction, intracellular transport, cytokinesis, endocytosis, exocytosis, a bunch of things that the body is doing to basically move stuff, whether it's we're moving our body, we're moving a muscle, we're moving a chemical here or there. That's what all those expenses are. So energy is the work of the body requires energy, requires some sort of a source. That energy production in the cells is the chemical process of breaking down the glucose, and the cell requires a constant supply of glucose and oxygen. Glucose and oxygen are transported by water, and water is the medium in which is inside the cell for the glucose and the oxygen to be put to work. So that's our three major building blocks of life, of daily living, is glucose, oxygen, and water. We've got to have them. So remember, metabolism is the process of offsetting the amount of energy created and the amount of energy used. Anabolism and catabolism. Anabolism, we said, is creating substances that are more complex. Catabolism is breaking them down. And then creation of the energy molecule, adenosine triphosphate. Adenosine triphosphate is what we hope to have when we break things down and put them into a simple substance that can release energy. ATP. ATP is a very efficient style of creating and excreting energy. It's usually done in an aerobic environment. So that aerobic environment means there's plenty of oxygen present to help it work. Now, where do we get some of these things? Sometimes we get some of our extra building blocks, some of our extra components from glycolysis. Glycolysis is glucose gets broken down into pyruvic acid, and pyruvic acid is really kind of our major gas, we'll call it, our fuel that gets used to create this extra energy in ATP. So aerobic metabolism is the complex process that results in a larger amount of ATP, and it also releases heat. So when we're talking about aerobic metabolism, we're going to create about 36 moles. That's kind of another odd concept, but 36 moles, units, we'll say, 36 units of energy during the process of aerobic metabolism. When we go to anaerobic, where we have less oxygen present, we really only create 2 moles. So it's 18 times more efficient to do it in the presence of oxygen than it is without. So there's a whole lot of product that gets lost when we do it without oxygen. Oxygen levels are not quickly restored. Obviously, the cell's going to die, and eventually this leads to the death of the person. Okay, so acid-base. Acid-base balance is something that our body is constantly in a state of doing. It's trying to say, how do I keep my pH, or my level of acid versus level of alkaline or base, how do I keep them most stable? So the body's constantly in a juggling game, a juggling process of keeping things on the up and up, keeping things stable so the body's in an ultimate environment to work. So think about it as the amount of heat versus the amount of cold. That if you're at 60, we'll say 65 to 75 degrees, most people are pretty stinking comfortable in that range. where there's going to be some people, we get above 75, they're considering it to be too stinking hot. There's some people below 65, believe it's too stinking cold. So it's going to be that constant juggling match to keep ourselves within that norm. So the acid base is used to is measured in pH, which means the power of hydrogen. So hydrogen, the acid, is a substance that can donate hydrogen. I have excessive amounts of hydrogen. I can give you some hydrogen. I have a high pH, which is actually a low number. Alkaline means I can accept some hydrogen. I don't have tons of hydrogen in my body or in my element, we'll say, and I would be happy to accept some, so I am a base. And when I have high base or high alkalinity, it is a high number on the pH scale. If you look at page 159 in your text, you will see the pH scale. It runs from 0 to 14. 0 is the most extreme acid we could ever possibly think of. You can see right there at 1, at a pH of 1, which is high acid, but it's low on the scale. High acid, stomach acid, which is hydrochloric acid. So very, very strong. We can work our way up. We can see things such as vinegar, tomatoes, saliva. Those things are slightly acid. Urine, that's the acidity. We get right in the middle there for a pH of 7, which is perfectly neutral. And neutral, the only real substance that we commonly know of that's perfectly neutral, is pure water. That's not with chlorine added to it. That's not with fluoride in it. That's pure water, usually done through the process of reverse osmosis in a perfectly sterile environment. Would give us that pure water. And it has a pH of 7. Our blood has a very narrow range in which the pH should be, and I'll throw this at you now. The normal body pH overall is 7.35 to 7.45. 7.35 to 7.45. So slightly basic, slightly alkaline. Basic and alkaline mean the same. We keep working our way down towards the other end here. We see things such as eggs are closer to 8, ocean water. So with that some salt water, we're talking slightly alkaline, between 8 and 9. Bleach is between 9 and 10. Ammonia between 11 and 12. And most of our real extreme oven cleaners are knocking on 14. So, which you may not have, that may be confusing to you. Well, they just work. These substances work in opposition to whatever it is that they're supposed to be working on. So if a majority of the filth and dirt that we're trying to clean something is acid, then using cleaners that are high bases causes them to have a better reaction, a stronger reaction. So if we tried to clean up, let's say we tried to clean up a tomato or grape juice, tried to clean it up with a cleaner with a pH of 4, we would see very little reaction. We try to clean it up now with bleach with a pH of 10. Those are farther apart on that scale. There's going to be a bigger reaction. So that's how pH can work. So acidic things, high concentration of hydrogen, low on the scale. And this is a negative logarithm, so if we want to talk more science there, a negative logarithm is why this is actually reverse of what you would commonly think. The buffer system is a system in which excess hydrogen ions are converted into substances that prevent acid accumulation, maintaining the acid and base balance. So when we buffer things, we're trying to basically neutralize them. So we may give something like bicarbonate. Bicarbonate is a buffer. Well, we have some bicarbonate around our house. A lot of times some of our antacids that we take are bicarbonates. Something like baking soda is a bicarbonate, and those things can be used to offset the strength of an acid. So they start to offset this acid, and in many cases, when you combine a buffer with an acid, say, or a base with an acid, when we combine those things, they'll create byproducts which are very easy for the body to deal with, like water, carbon dioxide. So the body can get rid of those things easily, where it's a little harder to get rid of acid specifically itself. Imagine trying to get rid of all of your excess stomach acid through either end of your intestinal tract. So, I mean, if you have to vomit and you vomit a lot of acid, what does it do? It burns all the tissues, creates lots of problems. So when if you would have diarrhea, obviously it can be problematic the other way. But if we can dump some antacids down there, or we can slow down the acid production, then we don't have to deal with acid leaking out those ways, and the body can get rid of the extra acid that it did have via urination and respiration. All right, membrane potential. So when we talk about membrane potentials, we're talking about a phenomenon that reflected by the difference in concentration of electrically charged ions on the inside and the outside of the cell. The electrical charge of the ions is determined by the ability to gain or lose electrons. So we're kind of getting back again to the cations versus anions. And this is, again, a balancing act. So if I have a lot of positively charged ions in one spot and a lot of negatively charged ions in another spot, we're going to see things happen, right? Think about magnets. Most people know there are positive and negative magnets. If you try to put two magnets of the same charge together, they won't. They repel each other. They just don't do much. But if you have a negative and a positively charged one, you try to put them together, sometimes they get together rather quickly and can potentially be a little forceful. It's the same sort of concept that we're talking about here with membrane potential. So we have an electrical current is that movement of ions that have too few or too many electrons, and the ions move from an area of greater abundance to an area of lesser abundance. So we'll take, for example, the sodium and the potassium. The potassium isn't as strong of a charge as the sodium is, so therefore the sodium can rush in through the various gated channels, and that sodium then gets pumped back out via the sodium-potassium pump, and the potassium back in, which requires that energy. But in just a normal flow of things, the potassium or sodium is going to rush in there because it has its bigger, badder, higher charge than we're dealing with potassium. So that's where we have this membrane potential. Remember, the sodium-potassium pump maintains those charges. And then we have polarized versus depolarized. So depolarization is when it stimulates an adjacent cell. A polarized cell is when it's ready to go. So when there's a difference between them, it's polarized. It's ready to do something. When it actually does that, it depolarizes, and then that in turn passes that signal on down the line to the next cell. So that's depolarization. That's what we have occur in the heart when a person's going to conduct an electrical charge down the line. One cell depolarizes, passes it on to the next cell, which is already polarized, so then it depolarizes, which passes it on next to the next polarized cell, and so on and so forth. All right, so think about it. The flow of ions across the membranes of a cell in the electrical conduction system of the heart is recorded in various ways with the electrocardiogram, or the EKG. That's actually what we're seeing, is those ions changing places. So the waveforms of the EKG or cardiac monitor provide information about the electrical activity of the heart. This is based a lot off of sensors and placing them in appropriate spots in order for us to know what is the norm. Now, when people don't have the normal flow of electricity through their heart, it changes the way that those waveforms look. So it may not have the nice normal look to it. So when the paramedics or the doctors or the nurses or whoever are watching that cardiac monitor, they're looking to see, does this have the normal waveform, compare it to what we know to be norm, or something look off? And we might compare it to a previous EKG they had, if we have one on file, to help us say, oh, okay, that's a little different than what they had before. Something must be not quite right. Something must be changing the flow of electricity here. And then we start to do a little bit more investigation. So an imbalance of electrolytes can affect the cardiac conduction. And how is this going to be reflected? Well, this is basically what I just said. So when electrolytes are off, we don't have as much maybe sodium as we would. So if we had a high level of sodium, it's going to rush in. If we have a low level of sodium, it doesn't move in quite as quickly. It's not as powerful because there's not as much strength in numbers there. So by having some of these electrolytes, and that's just an example of sodium, there's lots of electrolytes that are involved. Say we have a lower level of potassium. Same sort of thing would occur. It takes much longer for things to happen because we don't get the shift as quickly as we would if we have a very high-powered amount of that. All right.